This talk is actually going to be a little bit of 3 different talks, just enough to maybe get you interested in something, and then some ideas of where you can get additional information. I'm Robin Matthews. I'm the Director of Watershed Studies. I have been at Western since 1985, and I've been the Director of Watershed Studies since 1994. I actually graduated high school in 1970, which was, how many people know, the year that EPA was developed, all of those wonderful things. The Cuyahoga River had just started burning. Lake Erie was a toxic waste dump, and on and on. So I've seen a lot of it. There was no class called ecology. There was no class called environmental pollution back then. You kind of had to hide it. I once applied for a NSF grant when I wanted to be, I always said I studied dirty water. I applied ecology, it was a dirty word, so I studied dirty water. And I was told by NSF not to say that I was an applied ecologist because they didn't fund applied research. So things have changed. There are some things that are much better now. I don't think we have any rivers that are burning, although we do have tap water that will burn now, thanks to fracking. Okay, so I'm going to talk a little bit about what is this slime in my water. First, starting with a very, very brief history, like 2 slides, brief history of the Institute for Watershed Studies, which actually predates Huxley College. It was established in 1962 as the Institute for Freshwater Studies, and the major purpose at that time was to study Lake Whatcom. And if you're not local, you may not know that Lake Whatcom is the drinking water source for most of Bellingham and very large segments of the county, like in Sudden Valley. So when you're drinking water from the tap, even if you put it through a filter, you're drinking Lake Whatcom. And back in the 60s, there really wasn't much known about Lake Whatcom. So the primary mission was to study the lake. The Institute got some boats to go out and some equipment. And there was a little additional emphasis on regional lakes, streams, and groundwater. But most of the effort in the 60s and early 70s was Lake Whatcom. In 1982, so fast forwarding 20 years, nothing happened in between, fast forward 20 years, in 1982, it was renamed the Institute for Watershed Studies, recognizing that especially students at Western would have a broader interest than just that lake. The mission was expanded to emphasize looking at watersheds, still a freshwater focus, a little bit of marine, but mostly freshwater. But the idea that you really can't just take lakes and streams in isolation and look at them. In 1992, the Institute for Watershed Studies was certified by the Department of Ecology. And what this means is kind of twofold. First, it means that the data that are produced by our lab hold up against a private laboratory. So ecology will accept our values, even though they're generated by students, they'll accept the values as being equivalent to a private lab. The other thing is, man, that looks cool on your resume to say that you worked in a certified lab. Our current goals include, we're still monitoring the heck out of Lake Whatcom. We're out there monthly in the lake, and we go to a bunch of sites and so on. But a number of years ago, we decided, we being the staff in Watershed, decided that we really wanted more community engagement, not just Lake Whatcom. And so we started a public service, unfunded in other words, public service program that we called Small Lakes Program. It's now called the Northwest Lakes Program because it includes sites that are not really small. But we go out every summer and sample usually about 60 lakes in a bunch of counties, and I'll talk about that more. And then finally, last, but probably our biggest emphasis, is to provide freshwater research support for Western students, also faculty and staff, but mostly for students. If you wanted to do a project that involved freshwater research and you maybe needed a couple of samples run for nutrient analysis or something, we would help you with that. We can provide some equipment for the field. We don't have a lot of funding, so we can't do that, but we do have equipment, we have staff, we have training, and we will run small numbers of samples for free so that you can get your projects going. And we support a lot of undergraduate and graduate research projects. So I wanted to start with that first big project, the Lake Whatcom Monitoring Program, and just, this is really The annual funding for this runs between $250,000 to $350,000 a year, so it's a really big project, and I'm going to cram it into a couple slides. The emphasis is to conduct long-term lake and stream monitoring. It's the National Geographic approach. You just keep buying the same subscription year after year. The idea of long-term lake monitoring is that you want to maintain a very consistent approach to your lake monitoring so you can watch for trends. You can't do trend analysis if 5 years ago you changed to a different method and 10 years before that you had a different method. You have to have very consistent patterns. So that's still one of the biggest goals. The emphasis is on lake and storm event monitoring. Right now we're out in Austin, Euclid, and Silver Beach creeks when they rain, which I don't think it's going to rain again until fall. We also collect grabs from the tributaries, from about 10 tributaries around the lake. And we collect something called hydrologic data, which is at 15-minute intervals, how much water is passing down. We have gauges in 2 of the sites, Austin and Smith Creeks, which big water sources, and the other tributaries are monitored by USGS. So when we collect water samples, we measure them, we monitor the water, some cases in the field, some cases we bring samples back to the lab, and we measure a bunch of parameters that are kind of traditional, basic lake water quality parameters, nothing fancy. We don't do priority pollutants like, organic pesticides or anything like that. I've had students work on that, but we do, remember, long-term monitoring. So we do basic things like pH and temperature and the amount of dissolved oxygen. We also look at things in the lake itself, like chlorophyll. Chlorophyll is what makes algae green. And so it's an indirect measure for the amount of algae in lakes, and so on. So if you're not familiar with Lake Whatcom, it is a very interesting lake. It's a chain lake, glacial carved lake. It has 2 northern basins that we call Basin 1 and Basin 2. Each of those have sites kind of in the deepest part of that basin. Site 1, Basin 1, is adjacent to Blodell Donovan Park, if you're familiar with that. It's also, that's the part that's mostly in the city of Bellingham's limits. Those 2 basins are small. Each of them is about 2% of the total volume of the lake. They're shallow. They're about 20 meters or 60 feet deep. But they are the sites that have the most amount of residential development in their watersheds. And then there's a great big deep basin, and we figured 96% of the volume of the lake, so we decided that was worth 2 sites. We have Basin 3 at the north end, Site 3, and Basin 3, Site 4 at the south end. This is near Sudden Valley, if you're familiar with the lake itself. And as I said, it's 96% of the total volume of the lake, which if you're in a biostats class, you would say, okay, so that's the entire lake, right? Because statisticians, if it's 95, well, that's the whole lake. It's over 100 meters deep in many parts of that lake, which is over 300 feet, very, very deep. And all of the major water sources that come into the lake flow into that basin. So the places that we monitor, just to show you the map again, here's Basin 1, Site 1. City of Bellingham would be this way. Your city drinking water intake occurs here. Site 2 is right there, and then here are the other 2 sites, Site 3 and Site 4. So, teeny bit of lake ecology. During the winter, when it's cold, Lake Whatcom the water is cold from the surface to the bottom. The lake is called unstratified because if you measure temperature, it's almost uniform from the top to the bottom. My little blue diamonds are temperature, and that's about 7 degrees C, which is too cold to swim, let me tell you, don't fall in. It's cold. So this is Site 1, which is the farthest north, and you can see that it goes down to about 20 meters. And it's cold, and it's uniform. These are actual measurements from the field. This was February 2018. And students who like to work in the certified lab, I will say that it's a little hard to get students to go out in February. That's really not a nice time to sample the lake. Does anyone remember what it was like this February? I remember wading through snow to carry sample bottles out to the sampling crew. The oxygen and other things that are dissolved in the water, compounds like nutrients for algae, dissolved gases like oxygen and so on, because the water column is the same temperature from the top to the bottom, it mixes from the top to the bottom. Now this is only 20 meters or 60 feet, but even in Basin 3, where it's over 300 feet deep, it mixes from the top to the bottom. You'd see the same kind of straight up and down profiles for temperature and oxygen, okay? But an interesting thing happens in the summer. As the surface of the lake starts to heat, that mixing rate can't keep up with the heating rate. And so the lake begins to stratify. It stratifies into a warm surface layer and a cold bottom layer. And that stratification actually becomes very stable to the point that even a really strong windstorm cannot break stratification. And so once that happens and the lake is stratified, you'll see a temperature profile where it's warm at the top, still pretty cold, not quite 7, but it's still pretty cold at the bottom. And then you have a transition. And limnologists call this the epilimnion, which just means the upper part of the lake, and the hypo, like a hypodermic, hypolimnion, which is the bottom part of a lake. We like to use fancy scientific terms for things that are really kind of trivial. And the meta, as in between limnion, middle part of the lake. So epi, hypo, and meta is the transition. Once the lake stratifies, if you are in those northern shallow basins, the bottom of the lake, the hypolimnion, is isolated from the atmosphere. Now, an interesting thing, if you're not too familiar with ecology and photosynthesis, is that the primary source of oxygen in a lake is not from algae. It's mostly from the air, from the atmosphere. Algae do photosynthesize, and if you remember your 6th grade science, you know photosynthesis produces oxygen. So during the day, you'll often see little things like this, and that's actually a supersaturated band of supersaturated oxygen that if you pulled it up to the surface would bubble like a carbonated drink. It's supersaturated from algae at that level, pumping out so much oxygen that it actually is temporarily over the amount that the lake can hold, and it will eventually move back into equilibrium. It's just the rate of production is faster than the rate of equilibrium. So what we have up here is oxygen that is pretty much in contact with the atmosphere, so you get pretty much whatever the lake can hold at that temperature. In the middle here, if you have enough algae, you'll get, during the daytime, you'll get a little bulge where those algae are pumping out oxygen way too fast for the circulation to mix it. So, and then down here where it's not circulating, not mixing with the top, look what happens. The oxygen drops to 0. This is very much like a compost pile that you don't turn, if you're familiar with that. The reason you turn a compost pile is because you have organic matter, and bacteria are converting the leaves into soil, and in the process using up oxygen. Well, at the bottom of the lake, bacteria are chewing up dead leaf fragments and dead algae and so on, using up oxygen. But there's no oxygen down there because the algae that might produce oxygen are all up here. So what we see often in Basin 1 and 2 is very, very low oxygen at the bottom of the lake, like 0, okay? And this is something that progresses. So if you look at the blue line here, this was from last year. This is May, June, July, August, September. By October, the lake will start to cool again, and eventually a windstorm will come along, and the top and the bottom are close enough in temperature that it will mix. And then usually it stinks for a few days, and then all the oxygen mixes back in, and it looks like it did in the winter. But you can see that by September, it gets pretty bad down there. You have 0 oxygen almost through the entire hypolimnion. So this is my so what slide. Well, first of all, it's obvious. You have loss of habitat. Fish need at least 4 to 6 milligrams, and salmonids need even more, 4 to 6 milligrams of dissolved oxygen. So they cannot survive down at that level. You'll find them out around the edges where it's warm, but at least has oxygen. It also causes a release of nutrients and other things from the sediments. There are some like metals and methylated mercury and hydrogen sulfide that I could go into in more detail, but I won't. the one I want to focus on is phosphorus. So plants, when you fertilize them and you see something on a fertilizer that says like 5-5-5 or 10-10-10, that's usually phosphorus, nitrogen, and potassium for terrestrial plants. In lakes, what they need is mostly phosphorus and nitrogen, and especially phosphorus. Phosphorus determines how much algae is in a lake. Nitrogen and some of the other things will determine who is there, but the phosphorus determines how much. So we have these the sediments, as the oxygen drops, those sediments will start to release a lot of dissolved phosphorus that becomes available throughout the hypolimnion, and especially right up in this region here, which is why these bands occur. Those little bands of algae that occur right there, they are sucking up the phosphorus that is more available there than at the top of the lake. Okay, so that's a bad thing. And the other thing that was bad is that the rate, I actually got my slide out of order. There's the slide I wanted to say. The rate of oxygen loss in Lake Whatcom is accelerating, which means that after we stratify in May, it drops to 0 faster and faster. And you can especially see this. It used to be that by August, we still usually had a few milligrams of oxygen in most years, and now we don't. Similarly, in July, if you look at that, that's a really big change, where now sometimes we're all the way down to 0 in July. Because of the rate, the loss of oxygen, it's not just that there is no oxygen at the bottom of the lake, it's that the oxygen levels are dropping faster now than they were 15 years ago. That resulted in Lake Whatcom being placed on the state's 303D list, which is basically the dirty water list for the state, in 1998, due to declining oxygen levels in that hypolimnion. It was also placed there based on some problems with the tributaries. But from that 1998 placement on the 303D list, Lake Whatcom, the Department of Ecology developed something called a total daily maximum load for phosphorus entering the lake. And this is a binding document that the city and the county are having to work with in order to reduce the phosphorus entering the lake by 87%. They basically have to turn the developed areas, 87% of the developed areas, into phosphorus loading that resembles forest. Now, you don't have to take the development out, but you have to find a way to make development look like a forest and soak up phosphorus rather than dumping it into the lake. They have a 50-year plan for doing that. This TMDL was approved in 2016, and the city and the county are already instituting all kinds of protocols of methods for working on it. But the goal is to get that phosphorus loading level down so that we're not seeing a lot of increased algae. So how does this work? I told you more phosphorus equals more algae, okay? There are two main sources in a lake. It's real simple, internal and external, okay? Yes, there's some dry deposition, but internal and external. Internal is mostly controlled by this low oxygen that I was describing. So as the oxygen drops down, we start to get, in Basin 1 and 2, a lot of phosphorus being pumped out into the water column, and it's pulled into the algae that are right on that boundary between the surface and the bottom, okay? And that's what we were seeing here. But remember I said Basin 1 and 2 are only 2% each, so only 4% of the lake. Basin 3 is 96% of the lake, and yet it does not have this internal loading problem, but it is still showing a major increase in the amount of algae. So where's that coming from? Yeah. Coming from external sources in surface runoff. So this is the second part of that Lake Whatcom project, and that is these are all the creeks that we're measuring for both grab samples and stormwater. And if you look at the phosphorus in tributaries, these little red guys right here, region up here, which is the major residential area. And you can see that just from the grab samples that these are higher, these are intermediate, and these are your lower ones. So those are grab samples, but more important are the storm events. So grab samples, if you look at the range, we maybe get up around 100, but usually the median is around 20 to 30. Storm events carry an enormous amount of phosphorus into the lake. And here's, again, a little teeny bit of basic tributary ecology. The way we look at a storm event is by measuring, remember I said we had hydrographs? This blue line is 15-minute intervals over the course of 3 days. These little red dots are grab samples that we collect and then measure for, in this case, suspended solids, and in this case, phosphorus. So you can track it pretty well. Suspended solids are a good indicator of whether you have like exposed soil or impervious surface in your watershed. And those particles carry with them little bits of phosphorus. So when you look at a storm hydrograph and you see that here's where the flow peaked, you can see that the turbidity, the suspended solids, goes up with the storm, and so does the phosphorus. Okay. So you'll see that if you are familiar with what's going on in the Lake Whatcom watershed, you'll see that a huge amount of the effort to control phosphorus is focused on trying to treat or control or reduce stormwater runoff, because that's where an awful lot of that phosphorus is coming from that is causing the lake to increase in algae and then decrease in oxygen. This is my little picture that tells it all. This is actually a chamber that one of my students worked on. And on each side, you have stormwater. This right here is stormwater from the Lake Whatcom watershed. Okay. There's a membrane in between that allows enzymes to pass, but not algae cells. So on this side, we have phosphorus-starved algae. They're algae that are grown with not enough phosphorus to make them happy, and so they are really in bad shape. They want phosphorus. When that happens, they kick out enzymes that take phosphorus stuck on the surface of particles and rip it off, make it soluble, and that soluble phosphorus passes across that membrane and feeds the algae. So it's really good illustration that even stormwater that looks very clear will provide enough phosphorus for algal growth in the lake. So we have it all. We have internal sources producing phosphorus. We have external sources producing phosphorus. More phosphorus equals more algae. And not surprisingly, we see that showing up as increasing trends in the chlorophyll. We see it all through the lake. Site 1, 2, 3, and 4. All through the lake, we see increasing chlorophyll. Also increasing algae counts, but I'm trying to keep it short. So what this TMDL is going to try and do is reduce the algae, and in the process that reduces the organic matter that filters down into the bottom of the lake. It's at Basin 1, which in turn reduces, should help with that oxygen loss. If you have less food for the bacteria, you should slow down that oxygen loss. It's a really weird connection between algae, phosphorus, and low DO. So that's the goal. Hope we make it. If you want more information, I've included a number of sites. I produce annual reports with the rest of the watershed team. They're located on Western Cedar. We also have a Watershed Studies website that provides information. And if you want a summary of the TMDL for Lake Whatcom, it's available on Ecology's document website. So 20 minutes roughly for Lake Whatcom seems like enough. We wanted to do something that would be more community service. So that's our Northwest, AKA Small Lakes Monitoring Program. And we have a lot of sites. These are all the sites where we've collected data. It's a simple program. We just go out from a publicly accessible point, like a dock, and we collect samples using that nifty sampling pole and measure the data, run it through all of our certified analyses. We only do it once in the summer. As I said, we don't go out with a boat. We don't do anything hard. The students go out and collect from a bunch of different sites because it's not funded. So we try to maximize what we can collect. But you can click, if you go to the Institute's website, which I listed right back here, right here, you can click on this map, and if you click on any particular dot, it'll come up and it'll have you give you a little bit of information, like Canyon Lake. We sampled in Canyon Lake. And if you click on Canyon Lake, you can find it on the map. You can search for a link to algae that I've found in Canyon Lake. And you can also get water quality information from this site, okay? And this water quality information is very similar to what we collect for Lake Whatcom. From that, we have done dozens of offshoot projects, usually funded, small projects funded by community and consulting firms and so on. We've got one going right now for Lone Lake. I did one for Honeymoon Lake. We've done it for Padden. We've done it, a bunch of different lakes. Offshoot projects where we will go and try and answer specific questions. So for example, Honeymoon Lake. This was from last summer. Honeymoon Lake has horrible algae blooms, toxic algae blooms. And Honeymoon Lake happens to be one of the sites that we sample pretty regularly. Okay? So I was able to take the chlorophyll values and the phosphorus values and place them in something called a trophic category. So oligotrophic means not very productive. Those are the lakes that, when you look at them, are usually blue or clear. They don't have a lot of algae in them. We kind of like to swim in them. Maybe not the best for fishing, not always the best for fishing, but definitely nice looking lakes, very attractive recreational swimming lakes and so on. Mesotrophic, it's kind of the best of both because it tends to have good recreational access, fish and so on, but it doesn't usually develop nasty, horrible algae blooms. Eutrophic is the problem. You can have a pretty nice natural eutrophic lake, and that would just be where I'd go to find, yellow perch or bass or something like that. But a lot of eutrophic lakes are caused, the biological productivity is caused by runoff from agriculture or residential development or something. And so eutrophic lakes are the ones that tend to have toxic algae blooms. And what we're able to do is look at Honeymoon Lake and say, well, no wonder you're having a problem, sir. Your phosphorus is completely in the eutrophic range, provides plenty of food for algae. Your chlorophyll has been going up pretty much, and as a result, your trophic state is solidly in the eutrophic category. So the eutrophic category is yellow, and we see that pretty much for at least the last 5 years or so, we have had a eutrophic condition. And then I go in and look at the algae that are actually in it. And what I found, looking at the dominant species, were some of these fun Phanosomenon flossequi, Docospermum crassum, etc., fun taxonomic names. The 3 in yellow are potential toxin formers, which leads me to the last part. What's this slime in my lake? So this is Weiser Lake. Does anyone know where Weiser Lake is? Yeah, okay. Weiser Lake is kind of my go-to lake for toxic algae blooms. It has massive blooms. This is typical. This is not an atypical bloom at all. Massive blooms of toxin formers. I actually had a student looking at it every other week during last summer and measuring, among other things, algal toxins. And it was, for most of the summer, it was up in the toxic range. The neat thing about toxic algae is that it's a subset that's not too hard to identify. And so I've been working with community groups. I'm teaching an algae taxonomy class right now. We have the capability of measuring algal toxins, but what would be really nice is if we use our funding efficiently, because it's expensive to measure for toxins. Even the simplest test strip costs about $50 per sample. You can look at a sample using a $100 microscope from Amazon, and with a little bit of help, figure out whether it's likely, not whether it's toxic, but whether it's likely to be toxic. So the first question is, are they Cyanobacteria, which are blue-green algae, and those are the toxin formers in fresh water. The toxin formers in salt water are a different group, but in fresh water, it will be blue-green algae. So yes or no? And if yes, okay, is it toxic? So the final thing I wanted to mention was a public service where I've created a fairly simple online with my husband, computer science husband, online key, annotated because people don't want to have to learn a gazillion different terminology, terms, that will help you find out whether the species you're looking at is likely to be toxic. So for example, if we go here and you read things like filaments contain heterocysts. What the heck is a heterocyst? Well, they look like that. They're specialized cells that convert dissolved nitrogen gas into inorganic nitrogen that the cell can use. And if you have heterocysts, they're always these little round or oval things, kind of transparent, sometimes rectangular. And I've got pictures here so you can see what it looks like. And you just keep working your way down until you finally get to something that looks like what you're trying to identify. These guys are going to be some of the toxin formers right here. These guys right here. Okay. So you could take something like this and with a little bit of help go through the key characteristics. It's a colonial, irregular, or amorphous or spherical cell, colony with mottled dark brown cells that are not in a single layer, and that will take you to microcystis. And then this is microcystis, which is one of the major toxin-forming blue-greens. I try to annotate it and say, okay, that is different, and that is different. By the way, the zooplankton is fun. Those little zooplankton things are filter feeders, and they anchor to the blue-green colony because nobody's going to eat them. So to end, said 5:15 or 5:20? 5:20. Okay, because I got just a couple more things. So this cycles back to Lake Whatcom because in addition to watching the chlorophyll go up, one of the things that I've been watching that is an even stronger pattern is the inorganic nitrogen going down. Now, why is that scary? It's scary because high phosphorus with low inorganic nitrogen is the perfect environment for blue-greens. Most of these lakes that have toxic blue-green blooms will have high phosphorus or higher relative to higher phosphorus and low inorganic nitrogen. And that is exactly what I'm seeing in Lake Whatcom, which is a little scary. The good news is, yes, I mean, while there are lots of cyanobacteria, blue-green algae in Lake Whatcom, they're not the toxin formers. And that is a real nice thing. There's just tons of them. This is a bloom from Lake Whatcom, and it's got 1, 2, 3, 4 different species. And the only one that might conceivably be toxic is this tiny little thing called Pseudanabina, which is extremely rare, so wouldn't be producing enough to measure. All the rest of them don't produce toxins. Contrast that with Weiser Lake, and you have these guys. All of these are toxin formers. So if you want more information on that, I've listed, again, the IWS website, iNaturalist Guide, which has, so I worked with EPA to develop these things as well, and what you can do, you recognize some of these, is get a basic summary for each thing, the ecology, the toxicity, and lots of other information that's available. You can also click on any one of them and see annotations. Okay, and then finally I also linked to the state of Washington has an excellent website for toxic algae, this toxic freshwater algae. So I gave you a link for that as well. Okay, I think I have 2 minutes to spare. Yay.